This video is about cell parts, cell structure, and cell function. And before we talk about all the different parts of the cell, which are called organelles, I want to talk a little bit about the difference between prokaryotic cells and eukaryotic cells. This diagram up here is a diagram of a prokaryotic cell. All prokaryotic cells are bacteria, and all bacteria are prokaryotic. All other cell types are referred to as eukaryotic. I'll show you the spelling of that shortly. But this is a representative plant cell, and this would be a typical animal cell cross-section cut open so you can see the insides. Now, these are not to scale in terms of their size. Bacteria are much smaller than both types of eukaryotic cells, and typically plant cells are larger than the vast majority of animal cells. But I've made these enlarged so that we can see the individual inside parts. So prokaryotic cells, in addition to being smaller, are simpler in their structure. They only have a few parts. They have cytoplasm, which is the liquid inside that's mostly water. They have ribosomes, which are the site of protein synthesis, and the ribosomes in a prokaryotic cell do not have phospholipid bilayers surrounding them. They have DNA, and they have a cell wall with pili that help them stick to surfaces. And they also have one cell membrane. So the only cell membrane they have is on the outside of the cell, right inside the cell wall, and it is made out of a phospholipid bilayer. In contrast, eukaryotic cells have what we refer to as a true nucleus here and here. And included in that is a nuclear membrane that's separated from the rest of the cell by a phospholipid bilayer. In addition to that true nucleus with a nuclear membrane, there are many phospholipid bilayer surrounded or membrane-bound organelles within the cell, like chloroplasts and mitochondria and the Golgi bodies, and we're going to get into that more later. But the idea is, right now you should understand that all bacteria are prokaryotes and all plant and animal cells are eukaryotes, and that's also true of algae and fungi. So anything that isn't a bacterial cell is eukaryotic. So again, let's review the difference between prokaryotes and eukaryotes. Prokaryotes are bacteria. Eukaryotes are all other cell types. Prokaryotes have no true nucleus. They have DNA, but no nuclear membrane separating the DNA from the rest of the cell contents. Eukaryotes have nuclei with a nuclear membrane around the DNA. And prokaryotes have no membrane-surrounded organelles, and they only have ribosomes, and we refer to them as naked ribosomes because they do not have phospholipid bilayers surrounding them. And eukaryotes have many membrane-surrounded organelles. So now we're going to talk about the individual membrane-surrounded organelles that are in eukaryotic cells. So the highlighted portion of this diagram, the yellow portion, is the cell membrane. Sometimes you'll hear it referred to as the plasma membrane, sometimes you'll hear cell membrane. But it is semi-permeable, and you'll also hear the term selectively permeable. It means the same thing, and it controls what molecules will enter and exit the cell. Later in the unit, we will be talking about cell transport, and we'll talk about how different things can get in or out of a cell. The cell membrane is made up of two layers of phospholipids, a phospholipid bilayer, and some molecules can go across that lipid bilayer and others can't. And cell membranes are found in both plant and animal cells, and actually all cells, even bacteria. The bacteria, the only membrane that they have is the cell membrane on the outside. So this is a cross-section of a cell membrane showing you the phospholipid bilayer with the hydrophilic heads on the outside and the hydrophobic tails that have flipped towards each other away from the water. Water would be on the inside of the cell, but also on the outside of the cell. So all eukaryotic cells have membranes, lipid bilayers surrounding every organelle. The advantage is that the different chemicals needed for specific chemical reactions can be kept separate from each other in each organelle. This means the correct enzymes can meet the correct substrate molecules more easily. So now we're going to talk about the nucleus. The nucleus is referred to as the control center of the cell because it regulates cell activities. And the way it does that is by containing DNA, and you'll learn later next semester, how DNA controls all cell activities. DNA is the instructions to make RNA, which is the instructions to make proteins. So it controls all of the cell functions by having instructions and producing very specific proteins at the right time. So cell functions are all controlled by proteins, and the instructions to make them are in the nucleus. And nuclei are found in any cell that isn't a bacterial cell. So inside the nucleus is a material called chromatin, and chromatin is made of DNA, and it's a specific shape. There are two shapes of DNA, chromatin and chromosomes. The little X shapes that you usually see in diagrams are referred to as chromosomes. This looks a little bit more like a bowl of spaghetti or like a bunch of thread, and it is DNA, and DNA is found in every cell, even bacteria, although in bacteria it's just a single loop of DNA. But remember, DNA is made of nucleotides, and by dehydration synthesis we end up with a DNA molecule, that double helix. So chromatin is DNA. And of course, DNA is found in all cells, plant and animal cells included. So the nuclear membrane controls what goes in and out of the nucleus, and it keeps the DNA separate from the rest of the cell contents. And it's found in every cell except for bacteria. Bacteria have DNA, remember, but no nuclear membrane surrounding it. And the nuclear membrane is also made up of a phospholipid bilayer. So that little yellow structure inside the nucleus is called the nucleolus. Sometimes you'll hear it referred to as the nucleolus if you're watching a British or Australian video on YouTube. And it is where ribosomes are made, and it's found in both plant and animal cells. Now, these tiny little yellow dots that are all over the cell, some of them are floating around in the cytoplasm, and some of them are stuck on the surface of these tubes. Those tubes are called the ER. And those little dots, the smallest ones, are ribosomes. And they are where proteins are made. It's where amino acids join together to make a polypeptide, and where polypeptides fold into a three-dimensional shape. So we have our amino acid, our dipeptide, and our polypeptide, which folds into a protein. And all of that happens because of dehydration synthesis, and where that happens is at the ribosomes. Some of them are floating in the cytoplasm. Some of them are stuck to the surface of the endoplasmic reticulum, which we will talk about later in this video. Ribosomes are found in all cells. In bacteria, they have no phospholipid bilayers surrounding them, but in all other cell types, they have phospholipid bilayers surrounding them. So these yellow structures that are highlighted in here are the endoplasmic reticulum. And this is where lipids are made. The lipid components of the cell membrane are built at the ER. But in addition to that, the rough ER is the site of the completion of protein synthesis for proteins that will be exported from the cell. So if the cell is not making proteins that are going to leave the cell, the proteins are made on the ribosomes just floating around in the cytoplasm. If the cell is making proteins that are going to leave the cell, they start on the ribosomes in the cytoplasm, but then those ribosomes migrate to the surface of the endoplasmic reticulum, and then it will look rough because it's got all these little bumps on it, and then it's referred to as rough endoplasmic reticulum, or rough ER for short. And the endoplasmic reticulum is found in all cell types except for bacteria, including plant and animal cells. The cytoplasm, also called cytosol, is the watery fluid in the cell that's the site of many chemical reactions, and it's full of enzymes, and mostly it's water. And remember, water is polar and will dissolve a lot of things, so it's required for there to be water because it facilitates lots of chemical reactions that occur in that cytoplasm. So cytoplasm and cytosol are exactly the same thing. Sometimes there's more than one vocabulary word for the same structure in biology. And cytoplasm is found in both plant and animal cells, and in fact in every cell type. This is the Golgi Apparatus. It actually has 3 different names. It's sometimes called the Golgi Apparatus, sometimes it's called the Golgi Complex, and sometimes it's referred to as the Golgi Bodies, named after the scientist that discovered this organelle. One thing that's kind of tricky is that it looks fairly similar to the endoplasmic reticulum. It is just a system of tubes and canals. There are a couple ways that you can tell them apart. If you notice, the endoplasmic reticulum is a little bit more loose, and these are tight. And in addition, the Golgi apparatus is always closer to the cell membrane, whereas the ER goes all the way from the nucleus, usually all the way to the cell membrane. And the reason that it's close to the cell membrane is because it packages molecules for transport out of the cell. And most often, those molecules that it's packaging are proteins that are being excreted from the cell. And they are found in both plant and animal cells. Now, these yellow structures right here are bigger than the ribosomes, and they're like little phospholipid bubbles, and they're phospholipid bubbles filled with digestive enzymes. And they are referred to as lysosomes. Lys means destroy, some means body, so they're destroying bodies. Now, they have a couple different functions. They're for intracellular digestion of food, and they are the suicide sac for cell destruction. So sometimes they'll just open up and release enough enzymes to digest the entire cell from the inside out. And you might think that that's a bad idea, but in developmental biology, it's sometimes necessary. When a tadpole becomes a frog, it is the lysosomes that open up and digest the cells in the tail so that the tail disappears. And you actually, when you were an embryo, had a tail, and you had webbed fingers and toes, and those lysosomes open up and destroy those cells and change the structure of organisms. So it's very important. And in addition, it does help digest food. And we'll come back to that a little bit later. And lysosomes are found in both plants and animals, and all they are structurally is a phospholipid bilayer bubble filled with specific digestive enzymes. These yellow structures right here, that look kind of like a bean but with squigglies inside, are the mitochondria. Mitochondria is plural, mitochondrion is singular. You may see it both ways in writing, in textbooks and online. And mitochondria are the site of aerobic cellular respiration, and this is where we get energy out of our food molecules. And it produces molecules called ATP, which our cell uses directly to do work. So mitochondria are kind of interesting because they also have their own DNA and are about the size of bacteria and are very similar to some modern bacteria. So scientists have a lot of evidence to suggest that mitochondria were once free-living bacteria that became engulfed inside another cell. And these are found in both plants and animals. Even though plants make the sugar, they also need to break it down. So they make sugar from the energy in sunlight and then break it down later when they need it. ATP is the only molecule in the cell that can directly be used to do work. So this structure is called a food vacuole. And if you notice, there are in this diagram 3 lysosomes stuck to it. So the food would be there, the food particle would get ingested by the cell, and then the lysosomes would merge with it, releasing their digestive enzymes into the food vacuole, breaking down the food. So the food vacuole stores food and will merge with lysosomes to break down the food molecules into smaller molecules. And these are found in animal cells only. They are not found in anything that is photosynthetic, so they would not be found in plant cells or algae cells. Because they make their own food, they wouldn't need to eat and ingest things and then therefore digest them. This structure right here that looks kind of like an X, those are called centrioles. One is a centriole, and there are 2 of them in that diagram crossing each other, and they are referred to as centrioles, and they migrate so that one ends up on the opposite end of the cell during cell division. So centrioles produce spindle fibers, which are long fibrous proteins that during cell division pull chromosomes apart in order to separate DNA equally right before the cell divides. And these are not found in plant and algae cells, but are only found in animal cells. The cell wall is found in plant cells and algae cells and provides shape and structural support to plant cells. So in plant cells and in algae cells, the cell wall is made of cellulose. Remember, cellulose is a long strand of glucose molecules, and the oxygen atoms in the ring are alternating direction. So these glucose molecules are right side up, upside down, right side up, upside down. They alternate directions. Now, plant cells and algae cells are not the only type of cells that have cell walls, but they are the only type that have cell walls made of cellulose. Bacteria and fungi have cell walls, but they're made of different substances. Bacterial cell walls are made of a structure called peptidoglycan, and fungi have cell walls made of a structure called chitin. And chitin is actually the same structure that is found in the exoskeletons of crabs and lobsters and shrimp. So cellulose cell walls are found in plant and algae only, and not in animal cells and not in protozoans. But fungi and bacteria also have cell walls, though those are not made of cellulose. So this large yellow structure is called the large central vacuole, and it stores water and minerals and waste. And the water pressure, that's created because of this storing of water, makes it possible for plants to support heavy structures like leaves and flowers. So it gives them a little bit of extra support. And those are found in plant cells only. This green structure is called a chloroplast, and it is the site of photosynthesis. This is where light energy is captured and converted into chemical energy in the form of carbohydrates. So first glucose, maybe some sucrose would be made out of it in the case of sugar beets or sugar cane, and we could have starch certainly in corn and potatoes, and the cellulose for the cell walls. All of that comes from initially glucose that's made as a product of photosynthesis. And chloroplasts are found in plant cells and algae cells only, and they are always green. So these structures that I'm showing you in this diagram are called flagella or flagellum. One is a flagellum, and if a cell has more than one, they're referred to as flagella, and they are whip-like tails used for swimming, locomotion, and in this case the sperm needs to swim to the egg. This is a euglena, and the euglena swims towards food. The interesting thing about the euglena is that it is a heterotroph and an autotroph, so it has chloroplast and can photosynthesize, but it also can find food and eat. So it is sort of hard for scientists to classify because they don't know whether to classify it with algae or other protozoans that are heterotrophic. So these hair-like structures are also used for movement. In single-celled organisms, it's moved for movement of the entire cell. And in epithelial cells, like in the inside of our noses, and if you're female, your fallopian tubes, they actually move other objects across the surface. So they may move the whole cell or move objects across the surface of that epithelial tissue. So they're used for movement as well. So this is an amoeba, and those structures that are sticking out are referred to as pseudopodia. Pseudopodia is plural, pseudopod is singular, and there are two jobs for the pseudopodia. Pseudo means false, podia means foot, so it's false foot. So they're used to move, but they're also used to surround food particles. So they stretch slowly out around the food particle, and then the cytoplasm streams into it, and eventually these two pseudopodia would meet, and then there's a big food vacuole in there that would, of course, merge with lysosomes to digest that food particle. In addition to single-celled organisms that do this, you have white blood cells that engulf bacteria in a similar way to help fight infections and are part of your immune system. And they are referred to as phagocytes. And sometimes they're called macrophages. So pseudopodia are used for movement and used to surround and engulf food particles. So this is the end of our cell structure and function lecture.